This is a production of Cornell University.
Thank you, Jen, for the invitation. So actually, a couple of weeks ago, Jen talked to me about the seminar possibility here. I'm kind of scared because bacteria and the mammals and the plants are different kingdoms, right? And I thought, okay, well, we are from different kingdoms. How that can work here? But then I thought, okay, anyway, so the central dogma between plants and the bacteria and the mammals, they're the same, right? And in particular, also, I think plants also have the ubiquity. That's what feel like fine, we can do it. That's something here. And okay, here's my talk today. So we will concentrate to tell you about a story about how this bacteria hijack ubiquitin system in the host for the benefit of infections. Okay, so this is a bacteria called Legionella pneumophila, and you can look upon this structure of this bacteria is like a raw-like bacteria. It's a gram-negative intracellular pathogen. Actually, it's not like a replicated human pathogen, but sometimes it's a fresh host. Sorry, its natural host is a freshwater amoeba, and somehow when the people getting inhaled the aerosol contain this bacteria, and then it will cause a disease, an acute type of a pneumonia disease called Legionella pneumophila or Legionnaires disease. Okay, so now, so this disease actually first reported in 1976, I guess. So the Legion Convention in Philadelphia in the hotel, and a big ballroom, and 200-300 people in a party together. And after that, tons of people get sick. And eventually the CDC traced down what caused the disease. It turned out to be a bacteria. And that's why the Time magazine has this title here, Tracing the Philly Killer. And at that party, like 30 people died for this infections. And ever since then, because people are aware of the disease and have reported more and more cases every year. And there's a website actually, all cases reported to this website, and you can see a lot of it happens all over the world and every year, all the time. And I remember, you probably heard the news too, last year, Ithaca downtown has a kind of outbreak of the infections too, in Ithaca here. So now how they can cause the disease? So when the bacteria enter into a lungs, it will pick up by the alveolar macrophage cells through the process called phagocytosis. So as the bacteria goes into the cells, it has a weapon we call the type 4 secretion system. So you can see this needle penetrates the membrane, which engulfed encirculates this bacteria, and it delivers tons of bacterial proteins into the host, the cytosol. And then the magic happens. Basically, they just hijack a variety of host pathways, and they can intercept vesicles derived from ER and decorate on the surface of LCV. They secrete the proteins. They change the identity of these vacuoles, and then they can prevent the detections of by host innate immunity, like a fuse of endosomal lysosomal degradation. Not going to happen in this case because they change the identity. They hide the bacteria inside this vacuole, and then basically it expands and take the nutrients from the host, the bacteria replicate inside the vacuole within the cells, and eventually it will lyse the vacuole, lyse the cell, and spread the infections. So we do the infections in the lab, in tissue culture cells. You can incubate the bacteria with the tissue culture within 24 hours, almost like 2/3 of the cytocells will be occupied by this bacteria. And then it will lyse and break the cells and they spread out in bacteria. So, and as you can see very briefly, this brief like pathogenic pathways, it turned out if you look at it molecularly, oh, sorry, there's too many click. So those bacterial proteins secrete in the whole cells, they are sort of a better cell biologists than us. They know all the cellular mechanisms, cellular pathways. They secreted those weapons, and they can deal with hijack whatever variety of host pathways, including lipids, cytoskeleton, ubiquitin, you can name it. Everything in the cells, in the host eukaryotic cells, the bacteria knows how to manipulate and benefit for their growth. Okay. just give you some example, a taste. And initially we find actually this bacteria secreted this phosphatases, lipid phosphatases. And those like CDP and CDF, they can specifically hydrolyze those 3 species of the phosphatidylinositide, which in eukaryotes, maybe in plants, is the same thing. And it serves as a like zip code for different kind of organelles inside the cell. So initially, the phagosomes have decorated with the PI3P, 34, or 345, and then the PI3T is the marker for endosome. So you need to get rid of 3, and that's why the bacteria secrete those enzymes, remove all 3s, and eventually also other people reported as kinases here, and they convert all the phosphatidylinositide into PI4P, which enrich or the function as a marker for the for Golgi and ER type of organelles in the cell. And that's how they can hide this bacteria inside of the vacuole, now mimicking the host itself. And that's why the hosts don't detect this and just ignore this for degradation. So now, just to give you another example, to show you how the bacteria's knowledge of, they're so versatile and knows everything of the host. And this is another example. They also can exploit the host cytoskeletons probably for the entry or trafficking within the cell after they get into the cells. So there's an example here. So this is the effector called the MAVH. So it's very simple protein. It has a PI3P binding domains at the N terminus, and the C terminus has a WH-like actin binding motif. Very simple. However, when you add this liposomes contained incubated with these proteins and also added to the actins that is shown in blue here, you can see this actin was stimulated. This stimulated polymerization by this effector. And then you can see this not only polymerizes actins, it also deforms this membranes, tubulates. And it turned out that actually this effector secreted at a very early time during infections, once the bacteria make a contact with the host and recruit actins and polymerize actin at the contact sites and polymerize actin and speed up the phagocytosis process and facilitate bacterial entry into the host. So there's another example actually, which is right now actually my lab actually mainly focused on is that this bacteria also secrete numerous of effectors. They kind of muking or manipulated host mitochondria. So we know mitochondria is the energy hub and also produce a lot of small metabolites, which may be useful for the bacteria. That's why the bacteria needed to kind of hijack or utilize the host, rewiring the host mitochondrial functions for their benefit. And we did some kind of preliminary predictions, and we have like more than a dozen of those effectors. We probably they're localized, secreted and localized anchor to the host mitochondria and change the function of the mitochondria. And just to give you a little bit of example here. So usually this is the host mitochondria, what it looks like in eukaryotes. And if you have some, you will express this effector. For example, here is the LPG1803. It changes totally the morphology of mitochondria and causes the aggregation of mitochondria. And another example, for example, LAM12, it also secreted into the host, is anchored or localized to the host mitochondria. And when my student started this, and he found out that actually once this effector goes into cells, it recruits a host phosphatase to the mitochondria and dephosphorylates some proteins on the surface. And that's how they can inhibit mitophagy, basically stabilize mitochondria and inhibit mitophagy. So I don't have I don't have time to cover in details, but this just gives you a taste or some examples how the bacteria can invade a variety of host pathways. But certainly for today's talk, I will mainly focus on another major conserved pathway in eukaryotes, which is ubiquitin autophagy pathway. And again, the bacteria secrete numerous effectors and rewiring the host ubiquitin system and for the benefit of the growth inside intracellularly. Okay, so now we'll go this in more details. Before I give you some background here, so if we just look at the sequences of this bacterial genome, and you can identify some kind of a sequence homology, some effector actually did encoding the F box or U box, which are part of the ubiquitination machinery proteins like E3 ligases. And they can probably, some of them have been tested, actually, they can function as E3 ligases. On the other hand, there's some other effectors people can identify, and actually they can serve a function as the deubiquitinase. Okay, and so our lab actually are the first to also identify another unique type of hack-like E3 ligases, which represented by CDC and DCA. And I will tell you the stories about how we narrow down this as the E3 ligases and how they functions in the cell. Okay, so because as Jen introduced my research interest, initially I'm interested in phosphorinosis, lipid signaling and functions. And we started this because this protein, and it's a big protein secreted, and only people knows it combines the PI4P. And so the binding of the lipids is anchored on the surface of LCV, the Legionella-containing vacuole. And besides that, nobody knows. It's just really totally blank. So since I trained as a X-ray crystallographer, so we solved the structure of these proteins. It turned out that this protein has multiple domains, as you can see from here. And immediately from the structure, we can see a domain, which we call named the P4C. Actually, they combine the PI4P. Okay, now you can see so this P4C domain actually has a 4 alpha helical bundle. At the top of this is a pocket which is positively charged, which ideally can fit into this phosphorinoside 4P, 4-phosphate, which is negatively charged. And this actually turned out to be very specific. And we find this actually a lot of lab requested for this construct as a probe. You can use it as a probe to detect the PI4P in the cells. Okay, so this is the binding, but the N-terminus, we have no idea it is. This is the structure. And as a structural biologist, usually you will search the database and see which ones are similar to this, and then maybe you can infer any functional clues for this domain. It turned out this structure is unique, nothing similar to this. And we solved the structure. We have kind of struggled for like almost 2 years. I have no idea what it is, and it's hard for me to publish a paper without any function. So until one day, suddenly an idea come to us. So basically, we should analyze also the primary sequence to see whether or not there's any conserved amino acid, right? Because that's normally people do. But it turned out this protein actually, about 20–30% amino acids are sort of identical across different Legionella species. But you only see them in Legionella. You don't see the other species for this protein. That's why the homology is quite high between Legionella species. 20% is identical. And it's hard to see which one is important. But luckily, we have the structure. We can map those identical residues to the structure to see what happens, with the assumption that maybe this can function as an enzyme. And because if the function as an enzyme, then you should see a surface patch, which has a class of identical conserved residues. And indeed, excitingly, we see when we do that, we immediately see the pocket sort of a cluster on the surface here, which contained all the identical residues. Not in other places, or majority of them actually embedded inside the protein, which probably is for structural purpose. But only here you see the patch with conserved identical residues. If you look closely, and they have a aspartic histidine and cysteine. And I'm teaching biochemistry. Immediately I see this, I'm so excited. Okay, this must be a cysteine-based protease, or deubiquitinase, or whatever cell transferase, which I have no idea what it is. And then we order all different kits and tested in test tube to see whether or not we can detect any activity. But it turned out we cannot see any of those activity. So this kind of science just up and down, sometimes struggles, and that's one struggle, kind of another second hit for us, because we have no idea what it is. Then we thought, okay, maybe in the test tube, we're not really know the endogenous substrates. We don't know even the conditions of when we should make the function. So we should do it in the cell and ask the cell whatever something happens. So, and before we do that, we have a sort of idea, maybe it's related to ubiquitin. So we kind of co-transfer the cells with HA ubiquitin, either with the wild type or mutant proteins. And we mutated cysteine to adenine. We saw the cysteine must be important, right? And immediately, as you can see from the gel, we brought the Western blot with HA. to see the ubiquitin patterns. And you can see some band only appears in the wild type, but not in the mutant. And the mutant one, you see some band disappeared in the wild type. So that immediately tells you this effect must have something to do with the ubiquitination pathway, right? So with the help of my neighbor, Mark Smoka, they do mass spec and expert in mass spec. And we try to identify what are the changes, what, to ubiquitins and substrates, the bands, the protein changed. It turned out among the top 10 list, 7 of them actually relate to E1s and E2s. Okay, it turned out, so those in the cells, you always have this ubiquitin charge E2. And in the presence of CDC, this pool of ubiquitin charge E2 was diminished, disappeared. Okay, and that's why, and you can see if this part is gone, and that's why some endogenous ubiquitination was inhibited because you don't have a supply of ubiquitin charge E2. That's why some band suppose you should have, and now it disappeared. And instead, these enzymes or effectors, they hijack ubiquitin, take over ubiquitin from this E2s and go to the cell. And now it can recruit its own substrates, and the ubiquitin is its own substrate. Now from this diagram or hypothesis, you can see this actually functions as E3 ligase. It's hijack host ubiquitins and do its own ubiquitination. Now with this idea, it's much easier. You can now you have idea. You can test. You can confirm this during the test tube. So we can throw in the E1s, E2s, and the E3s here, and immediately you can see the robust ubiquitination happens with this E2s, but not with this E2, and with UBCG7, activity is so robust. Almost all ubiquitin used up within like 30 minutes, and also other E2s also functions fine. Okay, so now that tells you, wow, this is like a E3 ligase. And then it comes to the next question. So CDC should place here as a E3 ligase. So then we ask, this structure or protein like novel, how this can function as a E3 ligase? So basically we ask, how does CDC can recognize the E2s? Because the E3 need to recognize E2 and take over ubiquitin, and how they can transfer ubiquitin from E2 to its substrate or CDC. So we continued some mechanistic studies with the structural, solved a variety of structures, and you can see it turned out that there's a green domain here, and it specifically recognizes this E2, which is shown in pink here. And then through once it's bind to E2s, the ubiquitin, which is shown in yellow here, will be recruited to the active side, which is here, and transfer ubiquitin to the E3s and further on to its own substrates. Okay, so now with this, it turned out it sounds like this CDC or SDC, two of them actually paralogs in the same bacteria. So it is secreted, and from the fullness structure, actually, this active site is covered by the P4C domain, actually. So it's not active, probably. So then once it's secreted and sees the PI4P, and this P4C domain will bind the PI4P and opens up the active side, and maybe, and you can start ubiquitination some substrates on the surface or nearby. And we'll continue this later on. And before I do that, I will show you another ubiquitin kind of pathways here. So now as I go back to this here, so now I just present you our kind of identification of this HEC type E3 ligases. And recently, actually, there's another family of this effectors. And my collaborator, Zhaoqing Low's lab from Purdue, they find that actually this 4-member family of this CD family, it also hijacks host ubiquitins. But it hijacks ubiquitins through a very unique, bizarre way, as you can appreciate from the next slide. Okay, it turned out that this is CDE, the domain structure, and there's nothing that no ubiquitin ligase domains or anything like that. Instead, it has a ADP ribosyl transferase domain. And what it does actually, it specifically recognizes ubiquitin and modify ubiquitin, use NAD. And this process we call the phosphoribosylation. So NAD can be used as substrate and then attach phosphorybols, ADP ribose to ubiquitin, very specific and only to one residue, arginine 42. Very unique. Only ubiquitin, only arginine 42 will be modified by this domain. That's the effector. And then there's a second domain called the phosphodiester domain, PDE. What it does actually, it further modify or converts hydrolyzed AMP away from the ADP ribose, and what left is a phospholipase phosphate, and attach the phosphate to its substrate. And basically, you do this ubiquitination is very different from canonical lysine-based isopeptide linkage. Here is like a phosphoester linkage. So this ubiquitin now attached to the substrate, and also there's a phospholipase in bridge. So we work out the mechanisms and figure out how this enzyme can do this unique, we call it phosphoribosylate ubiquitination, PI ubiquitination. So as you can see from here, so this enzyme cleaves the phosphate and release AMP, and then the substrate comes in, activated by this histidine. So this is serine residues attack the phosphate, and now you can see the form of the linkage. So this ubiquitin now through the arginine, through the pentose and the phosphate attached to the serine residues on the substrate. So that's what we call the phosphoribosyl ubiquitination. Okay. And then it turned out that there's four of them, as I mentioned. So all of them can do the similar things, can do this, which is called phosphoribosylation. So, but if you look at the genomes of the bacteria, so there's other 5 effectors contain this conserved PDE domain. So we thought, okay, you look at sequences, all the catalytic important residues are all identical. We thought, okay, this PD domain maybe can do similar thing as do the ligations, as we just showed you in previous slide. So it turned out none of them can do the ligations. But surprisingly to us, it do the reverse. 2 of them can do the reverse, the PI ubiquitination, as you can see from appreciate from this experiment. So we can generate this phosphoribosylated species here. So this is substrate, this is phosphoribosylated bands. When you incubate this phosphoribosylated species with this DAPA B, you can see all the ubiquitin form is gone, and not the mutant. And similarly, so this DAPA A also incubated, you all cleave them up, use up, and not the mutant. So that tells you maybe this two actually function to reverse the reverse PI ubiquitination. And it also true during infections, as you can see from here. So we do the infections, you can see we can enrich the substrate, and in the wild type infection, you can detect some PI ubiquitin species. However, if you delete those 2 enzymes, now you can see this PI ubiquitin substrate really jumped high and then just substantially increased. Confirms this indeed function of the D-ubiquitinase, specific for this phosphor ubiquitination. To this point, you can appreciate actually this Legionella secrete those ligases and do this unique type of we call it phosphoribosyl ubiquitination. And they also secreted these DAPs to reverse the activity, to cleave these PI ubiquitins from the targets and recycle this. So just like canonical ubiquitination, you have the ligases and the DAPs, and the PI ubiquitin has similar pathways. To this point, actually, I have to confess, I'm so guilty for one of my former students, because we saw that this is really unique while we find this in the general species. It's hard to believe the nature only created this pathways or these cycles only in one species, not in other organisms or other life species. So we thought there must be some other lives have this too. And she spent quite a long time, but we never detect any of these enzymes in any other organisms. But maybe in plant, we have it, but we didn't really spend time on the plant species. So this is unique. It's really hard to believe only this Legionella have these pathways. So anyway, but this is not the end. So in top of this cycle, all this pathways, the cyclical pathways, there's another layer of complexity. So if you look at the genomes again, so this is the bacterial genome. We have the A's and B's and the C's. So those are the ligases I just present to you. Between the B and the C, there's 2 other genes nearby in between. So usually in bacteria, so if the genes form a cluster, their function is probably likely in the same pathway, right? They're probably regulated in the same regulon, whatever they express together. And indeed, if you look at what they are, is DAP-A, which is I just show you is the D-ubiquitinase. Okay, and another one called CDG. And interestingly, so CDG actually can rescue the suppressed activity of this ABC, the ligases, as you can see here. So this ligases, when you express the yeast and they are toxic, yeast don't grow. However, when you co-express CDG, now it regulates the toxic, rescue the toxic. So now yeast can grow. Just to cut it short, because there are too much stories behind this. So it turned out this again, we'll show the pathways. On top of this pathways, this CG, constitutes another regulatory pathways. Basically, the secreted and bind host comodulin and activate and modify CDE and inhibit activity of the CDE. So this we call it glutamination. And also there's other effectors that can recycle this. So basically we have this pathway, and on top of that, you also have a regulatory circuit to control the activity. And I think there's many labs contribute to this, including ours. This is really a new pathway, and that's why a lot of papers come out at almost at the same time. Okay, let's continue. So now to this point, I just introduced you to, we have this canonical ubiquitin pathways, and we have this unique PR ubiquitin pathways. And we want to ask whether or not these two different pathways, are they totally independent, or they can crosstalk with each other. The reason why we ask this is twofold, actually, because when doing infections, we do detect a lot of ubiquitin signals at the surface of LCV when you infected with wild-type bacteria. But now there's a mutant, and if you delete either CDE family or CC family, those ubiquitin positive vacuoles are significantly reduced. So basically, both of these two family ligases contributed to the ubiquitin signals at the surface of the LCV. And also, another fold of evidence reasons why we thought they're crosstalk each other, because they modify similar substrates. And both, for example, RAB1 has been shown can be both modified by the PR ubiquitination pathways and the canonical ubiquitination pathways too, as shown here. So you can see when doing infections, you can see this substrate being ubiquitinated. You delete either families, is reduced, delete both, it's almost all gone. Okay, so now we ask, if this function together and modify the same substrates, what kind of ubiquitin structures or chains accumulate or build up on the substrates? So we saw that here we come up with this kind of model. We saw that CDC can build up canonical ubiquitin chains, which directly attach to the substrates, and the CDE, they do this PI ubiquitination, and again, attach to the same substrates. And this also a ubiquitin molecule. Ubiquitin also has lysines available. They can be further ubiquitinated by CDC. That's why they probably can continue to build up elongated ubiquitin chain. And this type of ubiquitin kind of structures, we kind of hypothesize that maybe happens in the cell. And to test this model, we can do this in vitro in the test tube, but also in vivos in the cells. So you can enrich or accumulate this ubiquitin substrates, and then treat it with a canonical DAB, which will cleave all the isopeptide bond linked ubiquitin. So you free up ubiquitin, but only left this phospholipase ubiquitin attached to substrates, right? And you can also treat it with another enzyme, DAP-B, which I just showed you. It's a specific cleaves PRUB, the linkage. And then if you do that, now this ubiquitin will still maintain attached to the substrates, and then it will be released with a different lengths of ubiquitin chain. So now this is the kind of predictions, and we do the experiment and test whether this is true. So Marina synthesized this ubiquitin species with both enzymes, and indeed after treated with the DAB, you see the monoubiquitin, which is here, and some ubiquitin is maintained, which correspond to this substrate. And then if you treat with DAP-B, you see the monoubiquitin, diubiquitin, a little bit of poly, which correspond to this part, and you have more ubiquitin chains attached to the substrate, which correspond to this one. And if you treat it with both enzymes, you all collapse into monoubiquitin and almost all gone. So indeed, so those ubiquitin, you have two enzymes, they build up this kind of ubiquitin chain, mixed ubiquitin chain, we call it. And then in the cells, you have this mixed ubiquitin chain. And we know in the cells, we have autophagy machineries. We can detect those ubiquitins and send this for autophagy degradation. That's usually happening in the cell. And the bacteria is not that stupid. They know, actually, they know how to avoid this. But how? We don't know. Because this is ubiquitin, this should be recognized by ubiquitin adapters, and it can be degraded by the autophagy pathways. To ask this, we kind of have go back to the activity of these enzymes. So this can do the PI ubiquitin ligations, but sometimes in the absence of a substrate, in the presence of water molecules, actually do the cleavage too. So now, if this phospholipid was cleaved by water molecules, it will left a phospholipid attached to ubiquitin. So which means this ubiquitin probably be modified by CDE with phospholipid on arginine 42. And by these modifications, you may kind of mask the ubiquitins and block the recognition by the host ubiquitin adapters. And first, we'll verify this, whether or not this is true. This ubiquitin be modified. Indeed, we have ubiquitin chains synthesized, and then you can use we call it protein Q staining. You can only detect the free end phosphate attached to the proteins. And indeed, those ubiquitin chains are lights up, indicating in the presence of the SDA or CDE, indicating those indeed those ubiquitins be modified with pentose phosphate. And also, it's true in vivo and in vitro or in vivo both is true. When we use the ubiquitin substrates and we cleave it with a dot, and now we can analyze the ubiquitins. Again, those ubiquitins you can see in the presence of a CDE, which basically you can see this ubiquitin be modified. And then we also confirm it in mass spec, and this is true. So basically, so this ubiquitin be modified. That's why they can prevent the binding recognition by the ubiquitin adapters and prevent autophagy degradation. And indeed, if you look at the ubiquitin binding by this adapter proteins, the UBA domain bind ubiquitin and arginine 42 right at the interface. And you can imagine if the arginine 42 be modified with ADP ribose or phospholipase, and there's sterical collisions here, this block the binding. And indeed, when you do the binding assays, you can see if the ubiquitin be modified, there's no binding here. And you can otherwise, if without this ubiquitin actually can you pull down by the UBA domains. And also when you do the infections, it's too complicated, just focus on the two lanes here. So this ubiquitin species is a lot, but yet there's no P62 bind to this ubiquitin species. If you delete the CDE family, even though the ubiquitin species are being reduced dramatically, you can still see dramatic binding with P62. Okay, so, and this is also consistent with the imaging data. You can see what type of infections, no P62 be recruited to the LCV, but you delete the CDE, the ubiquitin, PP62 positive LCVs significantly increased here, as you can see. So, okay, I'm kind of speed it up a little bit here. So to this end, so now this actually, we understand now, so what kind of ubiquitin signal chains build up at the surface of LCV, and those ubiquitin actually is mixed, and also the ubiquitin moiety is being modified, and that's how they can prevent the host recognition, prevent autophagy degradation. But you may ask, why you need this? If you don't have ubiquitin, it's not gonna detect by autophagy anyway. So why you need this ubiquitin build up at the surface, and then modify them, prevent the recognition? So there must be some physiological functions associated with this ubiquitin, this mixed ubiquitin chain here, right? Indeed, actually, when we do the infections, if you look at the recruitment of ER material or ER vesicles to the LCV in a Y type, you can detect a lot of LCVs are positive for those ER materials, markers. If you delete either of these two family of the ligases, those ER markers will be reduced. Delete both of them, you can reduce it to almost to the dot A mutant, which totally block the secretion of the effector proteins. So that tells you, sounds like this ubiquitin chains you build up the surface also serves as a kind of a recruiting mechanism to recruit ER vesicles to the LCV. So basically, now if you go that to this model, so this, you can imagine this ubiquitin chain built up, and actually the ubiquitin within the chain, maybe they can use up to cross-link a second substrate through this phosphoribosylate linkage. If this ubiquitin attached to this first substrate, which anchor the LCV, and this one anchor an ER vesicles. So now you can imagine this ubiquitin chain serves as a anchoring molecules and bridge up together the two different proteins and bridge the vesicles recruited to the LCV. And okay, good. So I just cut in the short. Indeed, actually probably is true because we can detect those cross-linking of the two different substrates within the same ubiquitin chain. So you can basically, you can do the infections, you can pull down one substrate, you can also detect the second one. And then when you do the, when you do the cleavages, you can release the free second substrate in free. And indeed, when you do this assays, if you just focus on these two lanes here, so this, the ubiquitin species is also positive for the second substrates. And if you do the cleavage, you can see this second substrate that was released from the same material here, either with DAP A or DAB or canonical DAB or DAB B. So either one, you can release the second substrates from the ubiquitin species. Okay, so and certainly whether or not this ubiquitin chain serves as an anchor, still certainly need more evidence, more experiments. We're continuing investigated in this idea. So, but anyway, I will stop here and just summarize what I presented so far. And I told you this bacteria secrete many, many effectors, and they can really, they're the best cell biologists than us. And they know everything. They can manipulate the lipids and manipulate actin, cytoskeleton, and mitochondria, which I'm still working on. And I present you more in details about the two ubiquitin pathways. And these two ubiquitin pathways function together. They build up sort of a mixed ubiquitin chains, like a meshwork on the surface of LCV. Not only they can protect the bacteria from host degradation, but also probably can serve as this anchoring scaffold to recruit host materials or ER vesicles. And okay, by the end, I will see some people's, I acknowledge that I do see some of the former members and some new members, and I don't have time to go in details, sorry for that. And also some collaborators and the foundings, and I will stop here, and hopefully it's not too late, and welcome any questions.
Question. The first one is that you showed target RAP23.
Yeah.
Why is that really a target? Why is that a target? Like, do you have any idea like why RAP23 should be degraded as opposed to being a partner? Is there any other target for this effector? And do any of these proteins have any role towards 23.6S proteasome, or is this ubiquitination basically only works towards autophagy, like as a substrate for autophagy, and not for the proteasome?
Yeah, maybe I'm not clear with this. It turned out these ubiquitins are not for proteasome degradation. The purpose is not for degrade any proteins around LCV. Totally not for degradation. So in terms of substrates, we did a mass spec with both CDCs and SDAs. You will see a numerous of effectors, which many of these actually are ER proteins and some proteins involved in membrane trafficking, like RAP, small GDPases, and many others. And there's also some of the substrates that are kind of common for each ligases. And we believe there's no specificity for those targets. I think it's more likely based on the geometry, based on the space, the vicinity. So because those enzymes, when secreted, it's all localized on the surface of LCV. Whatever protein nearby, they can be modified. And the modification not for degradation, instead, they probably just build up ubiquitin mesh as a protecting shell, and also serve as I mentioned, as an anchoring scaffold and recruit other proteins, cross-linking other proteins. So does that answer you?
more membrane to that area?
Yeah, it's possible. That's why some initial vesicle can be recruited through other mechanisms, and they use this ubiquitin modified them and stabilize and anchor them more tightly through like a covalent mechanism. Just attach them together. Yeah. Yes, please.
That was fabulous. Mention the E3 ligase can serve as like a recruiter, or like the ubiquitin can serve as a recruiter. Why do Legionella do that? Like, why do they need ER to be there for that?
Why they recruit ER thing material? Yeah. So the bacteria need to grow, right? When it enter the cell, initially the phagosome is very tight. It's really tightly bounded only the bacteria size. Eventually, as I mentioned, 24 hours expanding enormously. Like 2/3 of cells are occupied by this vacuum. You need a membrane, you need materials to enlarge the space, enlarge the niche.
So it's more like a lipid transfer.
That's probably one of the mechanisms. Recruited ER vesicles, you can fuse them eventually, and then make it bigger.
Yeah.
And
Sorry, you see like some lipid transfer related protein around that kind of contact site or whatever?
yeah, that's a good idea. We never look at it yet, but maybe we have no idea yet. And nobody reported that any lipid transfer protein involved in this process or not. That's certainly could be a future direction for you can investigate. Yeah. Yes, please.
So when it replicates, it has cyclin and CDK, and the cyclins are ubiquitinated and then degraded, and that's how the cell progress, right? So if ubiquitins are hijacked, are the cell arrested in the same stage?
I'm not sure I understand quite well your question. Could you repeat it again? Or
The hijacking of this ubiquitination pathway
Mhm.
leads to cell arrest because cyclins, they will not be degraded.
oh, okay. I have no idea whether or not it interfere with other host ubiquitination pathways. It only so far what I present here is just utilize ubiquitin. It hijack host ubiquitin and use it for its own build up this ubiquitin meshwork. The cell cycle, the cycling or other ubiquitin pathways during infections, I don't know whether or not they, to what degree they impacted by this ubiquitin ligases. I have no idea. Yeah, and also the infection cycle is quite short, like 12 hours, you're done. And I'm not sure it's going to affect cell cycles or not. I have no idea.
Can we use,
Yeah,
can we use, let's say, these proteins
yeah.
that hijack the ubiquitin to, let's say, treat cancer or something? Because it will not let cyclins to degrade and the cell will remain in the same stage.
Yeah, that's a good idea. I always thought when I work on this, we call it weapons. These are really unique tools. You can use it to dissect the host cells. And but somehow those enzymes are quite toxic to the host. If you overexpress them, the host is not happy. The cell is going to die. So really, indeed, for example, this is really robust. If you overexpress too much in the cells, and just use up all ubiquitin and just hijack everything. And similarly, this ligase also too. You toxify ubiquitination system, endogenous ubiquitination. Yeah, it could be, if you use a small amount, you develop this as a tool, you can dissect, use it to manipulate it in different ways as you wish. Okay.
Fascinating talk. I'm
Thank you.
intrigued by the fact that the CID-C and CID-A are basically only found in Legionellas. What do you think their evolutionary origin is?
Yeah, usually people believe those genes, so the bacteria acquired from the original Haitian host. So we call it like a direct horizontal gene transfer. But now through evolution, this evolved in the whole bacteria, and now the sequence and all the other features maybe evolve some new ones, but they're totally different from eukaryotic host. Now it's totally different now. You cannot see any sequences, and even structure-wise, you don't see them anymore. Yeah, but how did nature created those? And whether or not, again, this is still puzzling me, whether or not maybe other species have this pathways or not, we still have no idea. And I know several labs try to investigate this, and nobody reported so far whether or not other species have those pathway or not. Maybe plant has it, I hope.
Yeah.
Okay.
pathogens.
Yeah, plant pathogen maybe also has it.
This has been a production of Cornell University, on the web at cornell.edu.